Hello everyone, welcome to another question answer session, and in this session we'll learn the GATB 2021 essentials, all the previous year questions from molecular biology section. So in this video you would get a detailed analytics of molecular biology topics which are important for GATB, and you can strategize your preparation based on these analytics. So stay tuned till the end of this video. But before that, you must be very confused at this point of time because you don't even know when the exam is going to happen and where to study from. You cannot stop your preparation for these examinations. You need to prepare for them because you need to think about your future, and you need a source from where you can learn MCQ, learn the concepts, clear your doubts, all of this. And that's why I would propose you might join Unacademy, because there are quite a lot of test series that are happening in Unacademy. For example, if you are going for CUCT, there is a test series for that. If you are going for BHU or DU, there is a test series for that. If you are preparing dedicatedly for GATB, there would be a test series for that as well. So all these test series you would get in one umbrella. That means it would give you a huge benefit for cracking examinations. So hurry up and enroll right now. So in order to enroll, you can download the app. You can use my code AP10 to get a 10% discount, and then you can start learning from India's best educator who would guide you throughout this process. You must be saying that what Unacademy gives you. In my opinion, it gives you the confidence to do it. It not only allows you to practice questions, clear your doubts, but it allows you, or it exposes you with all the leaders in the world. For example, Sachin is live on Unacademy in a specific day. So don't miss that out. It would give you enough motivation to do it. Learn about stories from the successful people and get confident. And that's what Unacademy provides you. Now, let's move on to the question answer. In JNU CEB 2018, JNU CEB was the previous name for GATB. So in JNU CEB 2018, there was a question which says the enzyme responsible for continuing DNA replication in prokaryotes once it is initiated is, this might be super easy for you, right? Options are polymerase beta, polymerase delta, polymerase 1, polymerase 3. So remember, they have talked about prokaryotes, not eukaryotes. So based on that, you have to frame your answer. Now, if you have chosen the answer, the right answer would be DNA pol 3. In prokaryotes, there would be DNA polymerase 3, right? Exactly. Okay, so you know DNA polymerase 3 can polymerize once there is a primer, and it would elongate from that 3' hydroxy end, and all of these things you know. If you by chance want to revise this concept, you can click on the link in the I button. You would get the detailed video of it. Okay, in the same year there was another question which says you are interested in a particular segment of rhinoceros DNA and would like to clone it in a cloning plasmid. You have following restriction map for the insert DNA and for the plasmid, and you have a tons of restriction enzyme choice. Question is, which of the following restriction enzyme would you choose to clone the DNA of interest into the cloning vector? So what would be the proper combination of restriction enzyme with which you would cut the vector as well as your insert, right? So you have to understand that part. If you have chosen your answer correctly, your correct answer would be X and N. Look here, X and N is just flanking the DNA segment of interest. X and N sites are also present in your multiple cloning site of your plasmid. So this justifies. Now, a counter argument is E and H. E and H is present in your insert as well as in your plasmid. But why not E and H? Because in case of E and H, if you use E and H combination, then you might lose a fragment of your DNA of interest, and that you don't want to do. So obviously you have to use the combination X and N, which is most feasible for you, right? Now you can also say that I would cut it with S. Now the S restriction site is present in the insert, but not present in the plasmid vector. So you cannot cut it with X. Okay, so this kind of question, this format of question is very important because they have asked a similar format in GATB 2020. I didn't put that question, my mistake. But anyway, this format is really important. Okay, another question. mRNA will form hybrids only with coding strand of DNA because DNA contains phosphate group at 5' end that will not reanneal at high temperature. So DNA contains hydroxy group at 3' end that will not reanneal at low temperature. RNA DNA hybridization follows the base pairing rules. Then denatured DNA is helical structure and will not reanneal to form hybrid, right? So you remember the process of transcription, right? So here mRNA would hybridize with the coding strand, right? This particular statement is not correct. So mRNA does not hybridize with the coding strand. It hybridizes with the non-coding strand or the template strand as shown in the figure. So this question was not at all proper, okay? And this hybridization occur because of complementarity and the base pairing rule. So overall, if this was non-coding or template strand, you can click on option C, but this question was a little bit offset in this particular year, okay? Fine. There is another important question that you need to know. Which of the following antibiotic is an inhibitor of RNA synthesis? So they can ask these possible type of questions like which antibiotic block RNA synthesis, which antibiotic block cell wall formation, which antibiotic perturbs let's say cell membrane integrity. So these kind of questions can be really important and you have to have that information. So here the correct option would be rifampicin. Now if you don't know how rifampicin work, you would get the link in the I button and it's very important. Okay, then there is a question in genetic code. A stop codon is a nucleotide triplet within messenger RNA that signals a termination of translation into proteins. Identify the correct pairs of termination codons. You have to just identify the stop codons from these options, and this is super easy. You can do it just like this. Okay, then question is, according to Chargaff's rule, the concentration of guanine always equals to that of concentration of cytosine. A segment of double-stranded DNA has 120 adenine and 120 cytosine, right? What would be the total number of nucleotides present in that segment, right? Exactly. So this Chargaff rule you have to know, and this is very easy. And if you have formulated the answer, this would be 480. I'll tell you. So adenine always pairs with thymine, right? AT and GC, this pairing is there, right? So obviously in a double-stranded DNA, you would have equal number of A's, equal number of T's. So if A's are 120, T's would be also 120. Okay, now we have 240. And if the cytosines, they are also 120, so the G's would be also 120. Now we have another 240. 240 plus 240 is 480. Which of the following antibiotics inhibit peptidyl transferase activity in bacterial protein synthesis? This kind of question, as I have mentioned earlier, is really important. So these are informative questions, but this is important, right? And the correct answer would be puromycin. Puromycin is specifically very important in context of like these kind of questions or exam point of view. Puromycin, rifampicin, alpha amanitin, these are really important. So keep a note of all of these. Okay, then there is a question which says Philadelphia chromosome is an abnormality associated with liver cancer, blood cancer, lung cancer, skin cancer. So let me tell you that this Philadelphia chromosome kind of topic is very important. Now here it would be associated with blood cancer. I'll tell you why. So what is Philadelphia chromosome? So this is nothing. It's a translocation between chromosome number 9 and chromosome number 22, and ABL gene comes closer to the B cell receptor gene, and B cell receptor is highly active in lymphocytes, and that is why this cancer is a cancer of lymphocytes. It's a cancer of blood cells, so it's a blood cancer, right? Okay. Then tunicamycin is an inhibitor of glycosylation, transcription, translation, fatty acid biosynthesis. So after solving many questions, what is clear to you by now is they have a affinity towards the functions of several antibiotics and inhibitors towards the central dogma process or other process. So keep a separate note about it. If you join Unacademy course, I have a dedicated notes which encompasses all of these things. Okay, glycosylation is the correct answer in this case. During synthesis of DNA by DNA polymerase, which of the following phosphate group of deoxynucleotide triphosphate is incorporated into the DNA? Very important question. Okay, very important question. This particular question is asked all the time. So the correct answer is alpha. Let me explain why. So here you see, this is a particular incoming DNTP, right? So this particular DNTP would get incorporated into the DNA strand, into the growing DNA strand. Now this particular alpha, which is known as, this particular phosphate, which is known as alpha phosphate, would be utilized to form the ester bond. This beta and gamma would not be utilized. This particular question can be formulated that, let's say you have a gamma 32 level phosphate or alpha 32 level phosphate. Now if you have gamma 32 level phosphate, it would not be incorporated in the DNA. But if you have a alpha 32 level phosphate, radioactive phosphate, it would be incorporated into the DNA and mark your DNA. So these kind of questions are very common in all the examinations, and this is one example, right? So here the key things that would be incorporated is this particular phosphate. Okay, in a DNA molecule, the guanosine content is 40%. Then adenine content would be, again, these kind of questions we have discussed earlier, and this would be really important. The correct answer would be 10%. How? Okay, so guanosine, G, right? G is 40%. So obviously cytosine will have to be 40% because G pairs with C. So 40, 80% is done. Now the total content of adenine and thymine would be in total 20%. Now since adenine number is equalized with thymine number, so there would be equal amount of A and T's. So you have to divide 20% with 2, and that is 10%. Okay, the mRNA is complementary to the nucleotide sequence of which of the following DNA? Coding strand, anti-template strand, sense strands, anti-sense strand. This is another question which was asked previously. So remember, this type of question is really important, right? So the mRNA sequence is exactly the sequence of the coding strand, except the U's in places of in places of T's. So obviously the template strand would be complementary to the mRNA, right? And template strand is also known as antisense strand. So here the correct option would be option D. Okay, moving on to another question. Substitution of thymine base by adenine in DNA is known as what type of mutation? Transposition, transition, transversion, frame shift. And if you know the correct answer, it would be transversion, right? So these kind of informative questions they do ask in GATB. Okay, a polypeptide has a molecular mass about 200,000. The most probable number of amino acid that this polypeptide has is these given numbers. How to go about this problem? The correct answer would be 2000, because roughly your amino acids, so that all these particular, you have to divide it with kind of 1000 and you would get an overall number. Okay, now EMS test is a rapid method for detection of oncovirus, retrovirus, chemical carcinogens, typhoid? The correct answer is chemical carcinogens. So EMS test tells us about the carcinogenic potential or mutagenic potential of a substance. If you don't know what EMS test is, you can quickly watch the video given in the I button. A major class of non-translated RNA function in the regulation of gene expression by base pairing with mRNA is ribosomal RNA, antisense RNA, regulatory RNA, small nuclear RNA. And these category of RNAs are known as regulatory RNAs. So regulatory RNA would be the correct answer. If you don't know about them, I have a video on that. VNTRs represent, we all know what VNTRs are. So they just ask the full form of VNTR, variable number of tandem repeats. Exactly. Expressed sequence tags are landmarks, okay, and they allows researcher to identify what? Okay, so it can allow the researcher to identify the start point of a intron, exon, etc. So expressed sequence tags are always important to understand. These expressed sequence tags means things that are expressed, okay? So obviously they would allow you to understand the beginning and the ending of an exon or beginning or ending of an intron. Okay, now another question was asked where they said codons that specify same amino acid often differ in the third base, right? This is wobble hypothesis. Exactly. C-value paradox tells us about linearity of the relationship between genomic size and complexity of the organism, non-linearity of the relationship between genomic size and complexity of organism, dosage compensation, number of chromosomes. So obviously C-value paradox give us an idea that there is a non-linear relationship between genomic size and complexity of organism. Many simple organism has complex genome than a complex organism. So this is actually C-value paradox. Okay. What function you might postulate for a polypeptide having a zinc finger motif? So it has role in signal transduction, transcription factor, growth hormone receptor, or cytoskeletal component. So the most probable answer here would be the transcription factor, because many transcription factors, especially the steroid receptors, they have zinc finger motifs which binds to specific DNA elements. Okay, now DNA fingerprinting has proved in forensic science. It involves the usage of many satellites, rRNA, cDNA, bacterial DNA. So which one would be the correct option regarding this? Wait a second. So the correct answer would be mini satellites. So mini satellites are analyzed in DNA fingerprinting. Okay. So just to give you an idea, let's say individual 1 and individual 2, there could be differences between their mini satellite pattern, and that pattern can be compared with some findings which is from a crime scene, let's say. So this is how DNA fingerprinting works. Then you talk about retrotransposons, transpose by, cut paste mechanism, copy paste mechanism, gene amplification mechanism, or gene deletion mechanism. And the correct answer is copy paste mechanism. They again asked a question. Okay, so let's talk about what is copy paste mechanism. So retrotransposon make a copy of that particular transposon and allow integration of that copy in a different genomic site. So it's not a cut paste, it's a copy paste. The initial position is retained. Short sequence of DNA used for identification of complementary sequences is called a probe, marker, aptamer, or express sequence tags. And the correct answer is probe. And this kind of probe you have seen used in like southern blot. Then it is used in situ hybridization and many other usage, right? So probes are really important when it comes to GATB. Okay, and if you want to know how probes are generated, how probes are leveled by radioactively or non-radioactively, you can click on the link in I button. Okay, in prokaryotes, the lagging strand primers are removed by DNA pol 1, pol 3, 3' to 5' exonuclease DNA ligase. And the correct answer is DNA polymerase 1. An enzyme that relieves torsional strain while double-stranded DNA is being unwound is known as DNA ligase, gyrase, relaxase, or helicase. Correct answer is basically DNA gyrase, and we know if this is this particular is a topological constraint in our DNA, and that can be removed with the help of these gyrase enzyme, and now you can see a relaxed form. Okay, which of the following modification leads to protein degradation? Methylation, acetylation, acylation, ubiquitinylation. So there is a ubiquitin proteasome mediated degradation pathway. So this pathway ensures that polyubiquitinated proteins are marked for degradation via proteasome. So ubiquitinylation is the correct answer. It would have been better if they have written polyubiquitinylation, because monoubiquitinylation is not a mark for degradation. It's always polyubiquitinylation which marks a protein for degradation. So it should have been more clear, but it's not. But anyway, the answer here based on the correct these options are, it would be ubiquitinylation. So I hope you get a sense that from which particular topics you need to prepare. I hope this video was useful. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. My code, my course is also there in Unacademy. You can use my code AP10 to get a 10% discount on my course, and all the necessary notes, all these informations, they are nicely segregated there. You can download from there, and it doesn't take a lot of money to subscribe, so you can consider subscription. You should always try before you buy. Okay, thank you guys.